Hello, this is part 1 of Chapter 6, Microbial Growth. This is an example of a bacteria, Serratia marcescens. It produces a red pigment. Learning objectives for this section are classify microbes into 5 groups on the basis of the preferred temperature range, identify how and why the pH of the culture media is controlled, explain the importance of osmotic pressure to microbial growth. So, just like we humans have a physical requirement for our pH, microbes also have physical requirements for their pH. We need a certain temperature, microbes need a certain temperature. Believe it or not, we need a certain osmotic pressure, right? A certain amount of solutes dissolved in our bloodstream. All right, and then just like we need certain foods, bacteria need certain foods as well. Carbon, nitrogen, sulfur, phosphorus, trace elements are things that we only need a little bit of. So usually those are kind of what we call our minerals. They need oxygen. And from the outside, organic growth factors that encourage growth. So I think everybody already knows that microbes grow at a certain temperature. If you think about your refrigerator, right? Why do we put things in the refrigerator? Because microbes have a minimum growth temperature. So if we keep our microbes below their temperature, they won't grow and spoil our food. Okay, there's an optimum growth temperature, which is the happy place. Okay, this is where microbes are going to do their best, fastest growth. And then there is a maximum growth temperature. All right, now with what we already know with our potato catalase experiment, we know if we get things too hot, what is going to happen to their enzymes? The enzymes are going to denature. So if we get microbes above their maximum growth, they are going to go ahead and just denature. Their proteins will be misfolded and they won't be able to do any chemical reactions. All right, so physical requirements. Some like it cold, some like it hot. Psychrophiles are cold-loving. Mesophiles like a medium temperature. Thermophiles like it hot. All right, so looking at this chart, knowing that human temperature is going to be a 37 degrees Celsius, that's approximately human temperature, which of these is going to be more likely to be a human pathogen? A psychrophile, a psychotroph, a mesophile, a thermophile, or a hyperthermophile? Which one do you choose? That's right, you got it. Mesophiles are going to be human pathogens because they like to grow at human temperature. So unfortunately, a lot of bacteria are mesophiles and they love to grow in our human bodies. All right, psychrophiles are going to grow about 0 degrees, optimum growth 15 degrees Celsius. That's cold. Psychotrophs can grow at 0 degrees Celsius, optimum between 20 and 30 degrees. And unfortunately, these guys are the ones that can live in our food in the fridge. All right, mesophiles are going to have their optimum growth 25 to 40 degrees. These are going to be the normal microbiota and unfortunately pathogens of animals, and animals includes us, humans. Okay, so a lot of what you're going to be doing as a nurse is to be getting rid of those pathogens that humans have. All right, what are we talking here? Temperatures for food safety. Okay, so very slow bacterial growth, over 60 degrees. The danger zone here, unfortunately, is pretty much room temperature. And then some can grow in the fridge. So when you're at a picnic or what have you, unfortunately your food is going to be in this danger zone. So don't leave food sit out too long, especially in Florida. Okay, so what are we looking at here? This is your temperature, and this is the number of hours that it's been left out. So refrigeration is a wonderful thing for preserving your food. We're looking at the approximate temperature range at which Bacillus cereus multiplies in rice. So I know a lot of people like to do a fried rice thing, reheat leftover rice. Is a bad idea in Florida. If it's been living outside of the fridge too long, you can get sick. Okay, again, thermophiles, optimum 50 to 60 degrees Celsius. So hot springs and organic compost piles. So some people like to throw their food outside to rot, and those thermophiles can live in there. Okay, hyperthermophiles, not likely to be human pathogens because they like to live over 80 degrees Celsius, so like volcanoes and things like that. All right, pH. We can actually use pH to preserve our food. I'm sure you've probably seen sauerkraut, pickles, cheese. All of those are going to ferment so that they become a little bit acidic. Okay, so bacteria like to grow between 6.5 and 7.5 pH. And unfortunately, remember our blood, 7.35 to 7.45. Now, unfortunately, this is the happy place of pH for bacteria. They'll be delighted to grow in your blood. Okay, we've got mold and yeast. They're going to be between 5 and 6. So unfortunately, that means they can grow in some of your foods. Okay, acidophiles grow best in acid, and we looked at buffers in our class, right? We used a buffer to keep our solution at a pH of 7. Our solution was plain old water, right? So water is neutral. It has a pH of 7. Neutral water, pure water, neutral, has a pH of 7. And in our classroom, we did do a little experiment where we put in a buffer solution and it kept our pH at 7. So sometimes if you're trying to grow something, you have to put in a buffer to keep the media where the bacteria or whatever you're trying to grow, yeast, could grow. All right, we're going to do an experiment this week coming up with osmotic pressure. Okay, we're going to get some algae and we're going to put different solutions on it, and we'll be able to see the plasmolysis. Plasmolysis. It's going to shrink the cytoplasm, right? Lyse. It's going to take the cytoplasm out. All right, now some microbes prefer a very high salt concentration, 30% sodium chloride. Extreme or obligate, hallow means salt. Phile means love. Halophile, salt loving. Facultative means it can go either way, okay? So it can tolerate high salt concentrations, but it doesn't have to have high salt concentrations. All right, so we'll be doing this experiment coming up this week, unless it pours and pours rain, because I'm going to get the algae out of the pond. So if it's just crazy raining, we might have to wait till next week. So algae, we're going to get some out of the pond, and it's very similar to bacteria in that it does have a plasma membrane and a cell wall. So it's easier to see under the microscope though. So we're going to create different solutions. You guys are going to create the solutions and you're going to make a hypothesis about what is going to happen to the cell, right? Will water be sucked out? Will water be drawn in? What's going to happen when we place it in different solutions? We can see a hypotonic. Hypotonic is going to have a lot of water. Okay, hypo means low, so the tonic is what's inside of the solution. Hypotonic means it doesn't have a lot dissolved in there. Now in contrast, hyper means a lot, right? If you're hyperactive, you just want to run around all day. Hypertonic means the tonic part is what's in the water. So the tonic is going to have a lot of solute. Hypertonic solution is going to have a lot of things dissolved in it, and we call the things dissolved solute. I remember it. You are in the bathtub, so the solute is what's in the water. All right, so why are hyperthermophiles that grow at temperatures above 100 degrees Celsius seemingly limited to ocean depths? Well, that's an easy one. Where are we going to find temperatures above 100 degrees Celsius? The liquid magma that's inside the earth, right? So volcanic vents, things like that are going to have that high temperature. Okay, other than controlling acidity, what is an advantage of using phosphate salts as buffers in growth media? Well, the microbes also need phosphate to grow. All right, next question. Before the development of modern food preservation techniques such as pasteurization and canning, why might people have used techniques that rely on osmotic pressure? Well, we learn now that it's not so good for you, but it's really easy to put salt or sugar on food to preserve it, right? Real easy process. Salt and sugar. Okay, those are two things that are pretty abundantly available, and you can just put them on there. So think about foods. What foods do we preserve with salt? Jerky. All right, so one way to preserve meat, smoke it, get it dried out, and then put salt on it. Okay, what about sugar? What about jam and jelly? Jams and jellies, right? So that is a way to preserve fruit. You put a bunch of sugar on it, and the sugar will suck out the moisture. Okay, so requirements for growth. What do microbes need to grow? They need the four main elements: carbon, nitrogen, those should look familiar, sulfur, and phosphorus. How do microbes grow? On the basis of oxygen requirements, and identify ways in which aerobes avoid damage by toxic forms of oxygen. Okay, so how does that work? Chemical requirements. Microbes do need carbon, as we learned in chapter 2 and 4. The structural backbone of organic molecules has carbon in it. All right, what is a chemoheterotroph? Okay, so here, troph means growth or nutrition. Growth or nutrition. Okay, here we're going to look at the energy source and the source of molecules to build with. Okay, so chemoheterotroph. Chemo means you have to eat chemicals, right? We as humans, we have to eat. Okay, we have to eat molecules. Our food, we're actually eating the molecules, and then we turn those molecules into our body. So microbes have to do the same thing. We have to eat molecules, and we use some of those molecules as the building blocks. Hetero means that we don't make our own. We have to use somebody else to make it for us. Okay, then we have autotrophs. Autotrophs are, for example, plants and some bacteria can use CO2. They get the CO2 out of the air. So I'm sure you heard of photosynthesis. Let's use green for the word photosynthesis. I'm sure you learned about this like in elementary school. So plants take the CO2 out of the air. They use photosynthesis to get the energy, and they stick the CO2 together to make their organic chemicals. Photosynthesis. Okay, so anything that's green is probably an autotroph, and it's taking the CO2 out of the air to make its organic molecules. Organic has carbon and hydrogen. Okay, so why do we need nitrogen? You know it already. You know, what is the structure of an amino acid? NCC, nitrogen, carbon, carbon. That's the backbone of your amino acids that are in your proteins. All right, you know what DNA is. You know it has nitrogen also. You have the nitrogen containing bases. Okay, and then ATP, it's just like DNA, right? Only it has 3 phosphates instead of just 1 phosphate. Okay, most bacteria are going to decompose protein-containing material for the nitrogen source. So grossly enough, sometimes that can be a human body that can decompose and rot by bacteria infection. Okay, so bacteria can also be our friends. Okay, they can get the nitrogen out of the air and use it for nitrogen fixation. Okay, because no matter how much we breathe, we can't take that nitrogen in. We don't have anything to do that. All right, then we have some bacteria are going to use ammonia or nitrate from organic material. All right, more chemicals, chemicals. Sulfur is used in amino acids, thiamine, biotin, and most bacteria are going to decompose the protein for the sulfur source. Some bacteria use sulfoxide or hydrogen sulfate. All right, phosphorus is found in DNA, RNA, right? Remember in class we had those little purple pluses that represented the phosphate group. Okay, so PO4, 1 phosphate, 3 oxygens is our phosphate group. It has a negative charge because DNA has a negative charge. Okay, so here it is, PO4, 3 minus. Okay, then we also have trace elements. So just very similar to humans, right? We have the same DNA molecules, ATCG. Inorganic elements does not have carbon and hydrogen. Okay, inorganic means it does not have both carbon and hydrogen. Okay, so some of these are part of our enzyme cofactor. We have a protein portion, and then we will have another portion stuck onto that protein that we call a cofactor. All right, trace elements include metals, copper, iron, molybdenum, and zinc. And often it can be in the tap water in the laboratory media. All right, next we're going to talk about gases. Gas. What is in the air? What do these microbes breathe, right? They are a little bit different than humans. Okay, some are just the same. They have to have oxygen. Obligate. Obligate means you have to. Aerobe refers to oxygen. You know, I'm sure you've heard of aerobics. If you're doing an aerobic workout, you have to breathe a lot of oxygen. Now, the word facultative means you can go with it or without it. Anaerobe, so they can either live with oxygen or without oxygen. Okay, if they grow without oxygen, that's called fermentation. Okay, anaerobic respiration is without oxygen. Fermentation, is with oxygen. Okay, so when there's not oxygen, they do anaerobic. When there is oxygen, they can do fermentation. All right, anaerobes and obligate, you could say this is an obligate anaerobe. Obligate, cannot have oxygen, it will die. Okay, so easy way to kill those is put oxygen on it. All right, aerotolerant anaerobes, they tolerate, but they don't use it to grow with. Then microaerophiles, they have to have a little bit lower than normal air. All right, a little bit less than the air contains, a little bit less oxygen. Okay, so why is this? Well, we've learned about what an enzyme is already, right? So whenever you use oxygen, you're going to be making what we call ROSs, ROS, reactive oxygen species. Okay, so there's several different molecules that contain oxygen that can actually react with your DNA and basically kill you. Okay, so believe it or not, oxygen breathing is not really necessarily good for you. Okay, so obligate aerobes, they're going to have two enzymes. We already learned about one because it's also present in potatoes. Okay, so catalase is one of those. All right, catalase. And then the other one is going to be SOD for short, superoxide dismutase. You see that ends in an ACE. Allows toxic forms of oxygen to be neutralized. And these guys can use oxygen because they have these two enzymes that are going to neutralize those reactive oxygen species. All right, next we have facultative anaerobes. So they can either grow with or without. You see this little extra bit right here, how it's a little more dense in microbes right here than it is down here, because they do grow better with oxygen, but they can also live without it, okay? So how do they tolerate those reactive oxygen species? Again, they're going to produce catalase. You guys already know that reaction. It's going to take hydrogen peroxide, H2O2, which can be damaging, right? You don't want that in your cell. And then it's going to go ahead and break it down into water and oxygen. All right, so that's what catalase does. And then superoxide dismutase is also going to do a reaction with oxygen that neutralizes the reactive oxygen species. Okay, what about this guy? Obligate anaerobes. Well, he does not have no catalase and no SOD, superoxide dismutase. Okay, so he has got to stay at the bottom of the test tube, right? We're just talking simple diffusion. Simple diffusion, oxygen is going to diffuse down the broth. So you're going to have, looking at these, we have high oxygen at the top, low oxygen at the bottom. I'm just using abbreviations. I know how to spell high and low, just to let you know. All right, so here we have aerotolerant anaerobes. These guys can live without it, but they can't use oxygen. So you see the difference between here and here. Right here we have a band where it has better growth. Over here it's just evenly distributed throughout. Okay, because this guy only has superoxide dismutase, so it can tolerate oxygen. It's not able to use it for growth. All right, then we have, see, this guy is a little bit down in the test tube where the oxygen is going to be less. Remember, the oxygen is just doing simple diffusion from the top to the bottom. All right, it's going to produce lethal amounts of toxic forms of oxygen if exposed to normal atmospheric oxygen. All right, so what are those toxic forms of oxygen? We can have a singlet oxygen, O2 minus, that has been boosted to a higher energy state and it is extremely reactive. So if you have a lot of this in the cell, it's just going to start reacting with all your molecules and really causing a problem. All right, then we have superoxide radicals. Okay, they are highly reactive and they can use or tolerate oxygen. And then we have that ones that can tolerate oxygen have superoxide dismutase. It's going to remove that superoxide and protect the cell. All right, then we have the peroxide anion. It's got a negative charge there. Okay, so we know that anions have a negative charge. Okay, the active component of hydrogen peroxide and benzoyl peroxide. All right, so the catalase enzyme, and here's one more enzyme, peroxidase enzyme, can remove that peroxide. All right, then we have hydroxyl radical. The OH is the most reactive free radical. All right, so what do these enzymes do? We've already learned this one, catalase. We played with it because it also is not only in microbes, it's also in potatoes. So we did this reaction in class. So just keep in mind, microbes also do the same reaction that our potato did. It's just more dramatic in a potato, it makes bigger bubbles. All right, so hydrogen peroxide breaking down into water and oxygen. All right, superoxide dismutase is going to take these reactive species, turn it into the hydrogen peroxide. So first this happens and then this happens, okay? Then we also have an enzyme. Notice it's ending in ASE, so it's a good guess it's an enzyme. It can take that hydrogen peroxide and turn it into good old water, neutral pH of 7. Okay, so this is what we did in class, except we used a potato. But here we're using a microbe. They just put the hydrogen peroxide onto the microbe. You see it bubbling. All right, so what other things do bacteria or other microbes need to grow? They need organic growth factors. So organic compounds that are obtained from the environment that contain carbon and hydrogen. Okay, so these can include vitamins, amino acids, purines, and pyrimidines. What was purines and pyrimidines again? My little way of remembering it is pyramids are sharp and they can cut you. So CUT are the pyrimidines. They're the nucleotides that have one ring. Okay, so cytosine, uracil, and thymine are the single ring nucleotides, and then purines are going to have a 2-ring structure. This is absolutely not it, but just to let you remember, I remember, it's corny, but if you're pure, you'll get 2 rings, a wedding ring and an engagement ring. That's just how I remember it. So purines are going to be the remaining nucleotides, adenine and guanine. Okay, so let's check this out. If bacterial cells have a sulfur source containing radioactive sulfur in their culture media, where are we going to find that radioactive sulfur? So sulfur is going to be found definitely in your proteins, right? Remember we have 2 amino acids that contain sulfur out of our 20. Okay, how could you tell if a microbe is a strict anaerobe? Well, that'd be super easy. You'd grow it with some oxygen and see if it dies. Okay, oxygen is so pervasive in the environment, it would be very difficult for a microbe to always avoid physical contact with oxygen. So therefore, what's the most obvious way for a microbe to avoid damage? What does a microbe have to protect it from bacteria? It has to have those, some of those enzymes at least, right? Enzymes that break down the reactive oxygen species. Okay, so those are going to include those 3 enzymes: superoxide dismutase, catalase, and peroxidase. Is there any way we can make that spell something, I don't know, SCP? Skip. Sort of spells skip, but not really. Superoxide dismutase, catalase, and peroxidase. Okay, so biofilms are gross and disgusting. Biofilms are formed from different types of bacteria growing together with a gooey, sugary glycocalyx on the outside. Okay, so microbial communities, different types of microbes living together, form a biofilm. So slime or hydrogels that adhere to surfaces. So biofilms are bad because they can help microbes stick on, adhere, stick on. That's the number one bad thing about biofilms. The number two bad thing is that it shelters bacteria from harmful environmental factors, including cleaning products and antibiotics if they're in your body. Cleaning products and antibiotics are going to be shielded by biofilms. Okay, so this is super interesting to me. Bacteria communicate cell to cell via quorum sensing. So when there's enough bacteria, they can actually change their gene expression, right? They can start transcribing different genes. That's pretty wild. So why should you wash your hands if the bacteria are just going to grow back? Well, you want to keep the numbers low so you don't have a quorum that will induce chemical signals so that other bacteria will be attracted. All right, so here's how biofilm works. These guys are planktonic. If you ever watched SpongeBob, you know one of the characters is called Plankton. So planktonic, he's all by himself. Little plankton is all by himself. So planktonic bacteria, there's only one. Then they're going to adhere to the surface, right? Remember adhesion. Adhesion is the number one thing for a bacteria to make you sick. Adhesion, adhesion. It sticks onto a surface or onto a human, right? Your teeth. This is how tartar gets on your teeth, as gross as it is. Okay, so then it's going to start secreting glycocalyx, which is a gooey stuff. And then there can be like water currents that go through, and then you get a whole big clump of, this would actually be like different types of bacteria, right? You're seeing some bacillus, but there could be some cocci in there, some round ones, all different kinds of bacteria can be in a biofilm. Okay, so outside of the body, they can clog up your sewage. They're also found in your digestive system. They're going to be 1,000 times more resistant to microbicides. Side means killing. So anything that says side on the end is a killer. Okay, and then 70% of infections are caused by biofilm. So you really want to take care of your teeth because believe it or not, if you have bad teeth, those bacteria are going to get into your bloodstream and they can get onto your heart valves. So dental caries, that means a cavity, right? A cavity is the same thing. You don't want to eat a lot of sugar because microbes eat the sugar and then they make acid that's going to dissolve your teeth. All right, catheters, uh, anything that's medical device that's stuck into a person, contact lenses, anything stuck on you can get biofilms of bacteria on it. So this is pretty nasty. This is Staphylococcus aureus biofilm growing in a catheter. So please do not be that nurse that's like, I'm an RN now. I don't have to help clean catheters. Well, you're going to be doing a huge disservice to your patient if you're not willing to clean, because this can grow on their catheter and get into their urethra and all the way up to their kidneys. So this is very bad. Boo, whoever let this happen. Okay, so a biofilm is beneficial to a pathogen. It's going to protect it from your immune system. Okay, protect from your immune system. And also, if it's outside of the body, it can protect from cleaners or anything like that. So protect it from your immune system. Immune system. I'm going to say immune response. So you don't really want a biofilm, right? That's why you're supposed to brush your teeth every single day, because otherwise you're going to get tartar on your teeth. What is tartar? It's a biofilm but with different bacteria in it. All right. Now, sometimes we do want to grow microbes and study them or determine what they are. So how do we grow microbes? Well, we got to feed it, right? How do you grow a dog? You got to feed it dog food. How do you grow bacteria? You got to feed it bacteria food. And we call the bacteria food media. Okay, so we have different types of media to grow different types of microbes, chemically defined or complex. Complex means, we don't really know exactly what's in it. Chemically defined, we know exactly what is in it. We know. All right, so we have different types of media: selective, differential, and enrichment media. And then we also have to, this is the food, right? Basically speaking, this is what type of food are we going to give it to grow it. Differential media, with your eyeball, you're going to see a difference. So you might see some media will turn red or yellow depending on what is growing in there. The pH will cause it to change colors, just like we used our universal indicator in class and it turned different colors. So some media will turn different colors according to what pH is in there, and different bacteria will cause it to be at a different pH. Okay, in addition, we just mentioned certain bacterias like different levels of oxygen. Some can't handle it at all. That's anaerobic, right? Some grow in the host cells, and some like to grow in CO2. So you can use a candle jar or you can use a bottle. These days in modern times, we use bottled CO2. All right, and then we're going to talk about different biosafety levels. I think these are going to be in part 2. But so most introductory labs is going to be level 1. Our lab, we do a few more advanced activities. So sometimes we're a little bit beyond basic in our class. We can go up to level 2 where you actually need to use PPE, personal protection equipment, personnel. All right, and then level 3, you're getting super dangerous. Level 4 is going to be things that are easily transmitted through the air that can be deadly, such as like Ebola. All right, so culture medium, we want to use sterile techniques. So sometimes we autoclave it. There should be no living microbes in our media. Okay, sterile, nothing alive. All right, so culture medium, like we said, the main thing, besides, of course, it can't have other microbes, is it has to have nutrients in it, right? You want to grow something alive, you have to feed it. Now, this word inoculum, we're going to do this in class. We're going to use some culture medium. We're going to use agar, and we're going to introduce some microbes from the environment into the media. So depending on what are you trying to see if somebody's sick, you're going to use inoculum maybe from their throat or their urine, right? And then you're going to culture it. So a culture is microbes growing in or on a culture medium. Okay, so we have different types of culture media depending on what kind of bacteria we're going to grow, or you're going to use a different type of culture media. Okay, so in our class, we are going to use some good old agar. Where did agar come from? Seaweed. Okay, so there was a scientist, and I think her name, I can't remember her name, but their last name, it was a married couple. She used seaweed agar to make like jams and jellies and things like that. So she got the idea, I'm sure she saw some stuff growing in it, that we can use this complex polysaccharide. What does that mean? Many sugars. Okay, many sugars. So we can use this, and then the microbes are gonna be able to live in it. Okay, so we can use it as a solidifying agent for culture media in Petri dishes or plates, slants, and deeps. Okay, so what is a slant? Basically, you're just gonna take a test tube and turn it sideways. All right, this is a test tube. Then you're going to put media in there like this. So it's going to be flat when you pour it in. And then we can make some in class. That might be fun. And then when you put it upright, it's going to have a slanted surface. Okay, so that's just going to give you like more surface area to grow your microbes on. Okay, so it's not usually broken down by microbes. So the cool part is it liquefies when you boil it, when you heat it up, and then it's going to solidify again at 40 degrees, which is about room temperature. So that's pretty neat. Okay, you heat it up and then you can put it in your petri dishes or your test tubes, and then it solidifies and you can put your microbes on it to grow. Okay, so let's talk about some different types of culture media. Culture media, things that you're going to grow it on. So one type, you know exactly what's in it. Chemically defined media, you know the exact chemical composition. So fastidious, I think of that as a picky eater. A fastidious microbe is what I call a picky eater. It's going to fast if you don't give it what it wants. Not really, but it's going to die because it has to have certain nutrient requirements. So fastidious, I remember it's a picky eater. It's going to fast if you don't have what it wants. It won't eat. So it has to have a lot of growth factors in the growth media or it won't grow. Okay, so chemically defined, we know exactly what's in it. Now, in contrast, complex media, complex, is going to be like just a bunch of weird stuff, almost like witch's brew or something. It's going to have extracts and digest of yeast, meat, brain, heart, plant, and so it's going to vary from batch to batch. So you don't know exactly what's in there. Examples are going to be nutrient broth and nutrient agar. Okay, so this is an example of a chemically defined media for growing a typical chemoheterotroph, such as your good old feces bacteria, E. coli. So some strains of E. coli are pathogenic, some are part of your normal microbiota or normal flora. So you see this one, we know exactly what constitutes the media. We know there's exactly 5 grams of glucose, 1 gram of ammonium phosphate monobasic, 5 grams of NaCl, sodium chloride, 0.2 grams of magnesium sulfate. This means that it has water with it. Okay, 1 gram of potassium phosphate, basic, and all of this is dissolved in 1 liter of water. So again, chemically defined means we know exactly what is in it. Okay, here's another example of a defined culture medium for growing Leuconostoc mesenteroides. Okay, so it has 25 grams of glucose, it has some salt, potassium phosphate, magnesium sulfate, and then a bunch of different amino acids. Okay, is this all of them? Alanine, arginine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. All right, and then it has some purines and pyrimidines. So this is where it's not 100% because it says 10 micrograms of each of adenine, guanine, uracil, and xanthine is a different amino or nucleotide. Okay, define culture for growing Leuconostoc mesenteroides. All right, it also has all this stuff in it, and then it Those phosphates serve as a buffer. Now, in contrast, this is a complex medium, meaning we do not know exactly what is in it. We know it has some peptone, which is a digested protein, but we don't know exactly which amino acids in it. Beef extract, it has some dead cow in there. We don't know exactly what's in the dead cow. All right, it just has some sodium chloride, agar, and water. Okay, then we have anaerobic growth media and methods for growing things without oxygen. Okay, so if you take something from somebody and you transport their culture in the oxygen and it's an anaerobic bacteria, it's just going to kill it, right? So if you're going to do a culture from somebody and you have an inkling that it might have some anaerobic bacteria in it, you're going to go ahead and try to grow it in an anaerobic condition. So some media contains chemicals such as sodium thioglycolate that's going to combine with oxygen to get the oxygen out, and then it can be heated to drive the oxygen out of the media so you can grow your anaerobic bacteria. All right, this is a jar that you might use something like this because that'd be more easier in a hospital situation, right? So this is a packet containing inorganic carbonate, activated carbon, ascorbic acid, and water. So when you put that packet in there, it's going to put off the CO2 so you can grow the anaerobic bacteria. All right, so this is an anaerobic chamber. It's got no oxygen in there, and you just reach your arms in there. All right, so then some things are really hard to grow. All right, so for example, Mycobacterium lepori, bacteria that causes leprosy. This is pretty gross, so don't touch armadillos. I don't know if you've ever smelled them, but ugh, a dead armadillo, it's growing bacteria that are very hard to grow. And then, as we mentioned in class, rickettsia and chlamydia, those are small bacteria that are going to be intracellular parasites, so they have to be grown in a tissue culture, right? So you'll have a petri dish or a flask, probably a flask, and in there you're going to have maybe human cells or animal cells, and you're going to be having inside of the individual cells, you're going to grow the bacteria inside the individual cells. All right, then we have what's known as capnophiles. This is a microorganism that's going to require more carbon dioxide for growth. All right, so again, there's a couple different ways we can do this. We can put a CO2 generating packet in an enclosed container. You can use a candle jar and actually burn a candle, and it's kind of the old-fashioned way. It's going to make CO2. And then you can also use a CO2 incubator that we saw in that previous slide, or it can just be a box that has a bottle with CO2, a tube with CO2 going into the enclosed little chamber, and then you're going to keep your bacteria in there.